Good afternoon, ladies and gentlemen. I am Dr. Udaya Cabral, attached to National Library of Sri Lanka. Today I present the work conducted by National Library of Sri Lanka. Uh, in addition to myself, two authors joined with me for this study. Professor Deepika Amarasinghe, University of Kelaniya, Sri Lanka, and the Dr. Pascal Querner, University of Natural Resources and Life Sciences, Austria. So, in this study, we could able to develop a very good control strategy for control library pest of libraries. The this study can be described as the effective, low-cost, simple heating box method to eradicate insect pest of libraries. First of all, I would like to give you some background information about the study, about the Sri Lanka and its environment conditions and the libraries and their facilities and services. So first slide, I will explain you that Sri Lanka and its environmental condition. Sri Lanka is a small island in the Indian Ocean. It is very close to the equator, just below the Indian Peninsula. This location creates a tropical environment on the island. Sri Lanka gets high rainfall up to 5,000 millimeter annually from two monsoons, southwest monsoon and northwest monsoon. The central part of the country is highlands. This area is very cooler than the lowland areas. So this area gets around 5,000 millimeter rainfall for the annually, and this area is rise around 5,000 millimeter from the sea level, and the annual temperature varies from 26 Celsius to 30 Celsius throughout the country, and the high humidity also very high, around 85% to 90% annually throughout the country. The dry season and wet season are obvious. There are no other seasons. So the wet season, especially for the rainy seasons, and the dry season. The temperature and relative humidity conditions throughout the year are favorable for insect growth and the rapid spread in the environment. So this is the background of the Sri Lanka and its environment. And the libraries. So we have around 5,000 libraries. We have good quality library service, but the thing is the building maintenance are not up to in minimum standards. There are many reasons for that. So we have many types of libraries, such as school libraries, public libraries, university libraries, special libraries, departmental libraries, several types of libraries. Majority are public libraries and school libraries. Library collections don't have proper environmental control systems or air condition systems or the dehumidifier systems to control the environmental parameters, temperature, humidity, like those things. And lack of trained staff also a challenge. And integrated pest management concept also introduced very recently for a library sector, but we use this integrated pest management some concept for the agriculture, especially the paddy agriculture in the country. So the integrated pest management practices recently introduced for the library and museum sectors in Sri Lanka. So the lack of knowledge about integrated pest management also the one reason for spreading the insects inside the libraries. We found several types of pests in the libraries across the country. The common and the important pest is order Sigentoma, or everybody knows about the silverfish. Silverfish is very common in the tropical areas such as Sri Lanka. So the very common silverfish is found in the library is Lepisma saccharina, and the Tenolepisma nigra also very common across the country, and Tenolepisma longicudata also found some places. And other important order is order Coleoptera. Order Coleoptera is very important because their eating patterns are very dangerous for the libraries. And Lesiodoma sericon is a very common species found in Sri Lanka, and the Stagopanizium also very common. We can find these two species everywhere in the country, and the Gastrolus and Indicus and Anthrus colorado especially find in the wet area, the wetlands in the country. Other important insects is order Isoptera. So we recorded two families in the libraries, Calottomidae and the Rhinotomidae. So very important one is family Rhinotomidae, Octoterms species. They are very common in the libraries, especially in the wet area. So their attacks is very severe and very difficult to control. So this is very important pest in Sri Lanka for when you're talking about library pest control strategies. So at the moment in the Sri Lanka we have no many options to control insect pests. We have several household chemicals available in the market, and especially we have phosphine chemical pesticide, especially we use for the agricultural and the agricultural crops storage purposes. So this is this phosphine is used for the libraries as well. The other traditional pest control methods also followed by many libraries, which is very common in Sri Lanka. We have traditional pest control methods came from our ancestors. We have a special plants extractions called a dummala. So it came from palm leaf manuscripts preservation. So there are many traditional pest control measures. Some are repellent, some are insecticide. So sometimes in many cases, these traditional pest control methods can't control huge severe infestation in libraries. In that cases, National Library of Sri Lanka help other state libraries to control insect pests in their particular collections. So in the National Library, we use phosphine fumigation to control and eradicate insect pest issues in all libraries. So it is very toxic and very difficult to apply in everywhere in the country because of its limit issues with the chemical. So National Library is planning to develop a new strategy to control insect pest. So with the aim of develop a new strategy to introduce for the rural libraries, this study is work out. So the research problem is there's no simple user and environment friendly economical feasible control method that can be implemented in rural libraries in Sri Lanka at the present. This is the research problem. Actually, we don't have good insect control method to introduce for the rural libraries in Sri Lanka. So to address the issue, we designed this study. The hypothesis of the study was insect adult and their immature life stages associated with library collections can be killed when they are exposed to solar heat. So solar is heat is we can get throughout the year, so it's very good energy source. So this hypothesis is that The objective of the study is to investigate a simple, user and environment-friendly and economical feasible insect control method for rural libraries in Sri Lanka. So the methodology, uh, for the methodology, uh, the first step was to a common rigiform box available in the market converted into thermal insulation box by wrapping black color water-based paint on outer surface in 2 times, and a data logger to read temperature and relative humidity was fixed to outer surface of the box. One prop was inserted inside the box while the other was kept outside. So we can measure the temperature inside the box as well as the outside. Temperature and the relative humidity also in the outside inside can be measured by this data logger. So the experimental process, the preparation of samples, the insect pests were obtained from cultures maintained by National Library of Sri Lanka. There are 3 types of species were collected for the study: Amarasinghe sericon and the Lepisma saccharina, Coptodomus species. So the adults selected for the from the Lepisma saccharina, and the larva and the adults species of Amarasinghe sericon were selected. And the Coptodomus species, we selected workers and the soldiers. So the preparation of bioassay, non-pest works introduced to packify all books and wrapped by a blotting paper. 6 replicates were prepared as experimental samples. Control samples were kept in room condition. Room conditions mean the temperature around 28 to 30 and the relative humidity is 85 to 90. pH was measured because pH is very important for the permanence of paper materials. So we can find out any effect from the also kept in the outside as a control samples and also kept with the experiment. So the rigiform box with the data logger was placed in the courtyard of the National Library on a sunny day for 1 hour. So when the inside temperature reached to 45 Celsius, replicate wrapped books were placed in the box. With a layout arrangement. The lid was closed tightly. The temperature and humidity levels inside the box are monitored. So then 20 minutes, we obtained all the data mortality of the insect pest. So again, the experiment was repeated for 40 minutes and the 60 minutes. So Lepisma saccharina showed 100% mortality after 20 minutes exposed to temperature 45 Celsius. When the exposure time was increased to 40 minutes, the mortality rates of both adult and larva of Amarasinghe sericon and Coptodomus species were increased. The maximum mortality rate was 93 to 96 after 60 minutes. The results revealed that the experimental level of thermal increase has not caused pH in any paper materials. It is very important. And the discussion and conclusion, insect can survive in temperature as high as 60 Celsius as they are able to adapt to slowly changing temperature. The success of thermal pest eradication relies on rapidly reaching killing temperature so that the insects don't have a limit to adapt. Several scholars mention methods of thermal pest eradication involve a rise from room temperature to 52 Celsius within 1 hour or 4 hours. The thermal solution technique introduced by Dr. Piniga in 1996, was a technical refined version of the heat treatment. Not like the previous studies, the book in these studies are kept under solar heat only very short period, since the books are placed only after the inside box temperature reached to 45 Celsius. This method is safer than previous developed solar heating methods. So the blotting paper sheets used to wrap books in this method was to buffer the moisture fluctuation and to prevent moisture losses. And to prevent using the light fitting lid of the rigiform box to help to minimize the interior temperature and screen out UV and visible light, which are harmful to paper materials. 100% mortality of Lepisma saccharina was within 20 minutes, 93 to 96 mortality of both larvae and adults of Lesioderma silicon, and 93 to 96 mortality of Copteroma species after 60 minutes were achieved using the solar heating chamber. Therefore, the solar heating method developed in this study can be simply used in rural libraries in the country during the dry season. Thank you very much for your attention.